Good morning, everyone. So, today let's discuss the topic mutation, next part of the chapter. So, what is actually mutation? Mutation means it is a sudden hereditary change, okay, due to which there will be variations occurring in the organism, okay. So, in very simple terms, we can say the change in hereditary unit, that is sudden change, is called mutation. Now the term mutation was actually first coined by Hugo de Vries, okay. In evolution part we have actually studied about the experiment of Vries in Evening Primrose, isn't it, regarding the evolutionary part. So he first actually termed the word mutation. Now basically mutation is of two types. One is the germinal mutation and other is the somatic mutation. The germinal mutation, it occurs in germ cells, okay, and these are actually, germ cells means the reproductive cells, and they are passed on from one generation to the next. That is, the parents transfer these germinal cells to the offspring, so this mutation is transmitted. But the somatic mutation, it occurs in the body cells. So when the organism die, simultaneously these changes also finished up or dies up, okay. So that means somatic mutation is not inherited, whereas germinal mutation is inherited. Again, depending upon the cause of mutation, it is again of two types. One is chromosomal mutation and other is gene mutation. Chromosomal mutation means the changes that will occur in the number or structure of chromosomes, okay, and gene mutation means the changes that will occur strictly in the gene of a particular chromosome. Now if the number is changed in the chromosome, then according to the change in number of chromosome, we find two types of chromosomal mutation: polyploidy and aneuploidy, okay. Polyploidy again divided into autopolyploidy, allopolyploidy, allopolyploidy. Then aneuploidy is divided into hyperploidy, hypoploidy. Then based on the change in structure of chromosome, we find the intrachromosomal and interchromosomal aberrations, okay. These are the different types. Then gene mutation. Now this is the basic actually classification division of the different types of mutation. So now let us discuss in details about the different types of mutation that are occurring. Okay, first the chromosomal mutation. That means the change in number and structure of chromosome involves the chromosomal mutation. So based on that, it is divided again into two types: the genomic mutation, which involves the changes in number of chromosome, and the chromosomal aberrations. Now genomic mutation is again divided into polyploidy and aneuploidy, okay. Polyploidy means the phenomenon of having more than two sets of chromosome. That means now triploid, tetraploid, hexaploid, isn't it? So that is called polyploidy. That means more than two sets of chromosomes are present in the offspring. Now sometimes what happens during anaphase, the chromosomal separation fails, okay. So when chromosomal separation doesn't occur, that means what happened, they will remain together forming an extra pair. So that is the result is polyploidy, okay. But artificially also we can induce polyploidy by application of colchicine and granosins. These are actually chemicals with the help of which we can multiply the chromosome number, that is double the chromosome number. Now it is divided into three different types: autopolyploidy, allopolyploidy and autoallopolyploidy. If the polyploidy takes place, or that means the increase in chromosome number takes place in the same genome, then it is known as autopolyploidy. Now you see here AAA, that means the same genome is been added. AAAA, 4 A's are there, that means same genome added. So this type of polyploidy is known as autopolyploidy. But if two different species are double after hybridization, then it is known as allopolyploidy. For example, see AABB. These individual is formed by hybridization of two different individuals. So this is allopolyploidy. For example, Raphanobrassica and Tritical. Tritical is the first man-made cereal, okay. So this is important. It is an allopolyploidy and it is the first man-made cereal. Raphanobrassica, it is crossed between two different plants and a new type of variety is being produced. Then auto-allopolyploidy means actually this is a allopolyploidy in which more than one diploid state is, that means one particular genome will have the diploid state. Now for example, see here AAAABB, that means AA is having its diploid state, okay, only one genome is increasing the number. So this type of condition is known as auto-allopolyploidy. Got it? The next is the aneuploidy, okay. After polyploidy we get aneuploidy. Aneuploidy actually it is the condition of having a few or extra chromosome than the normal genome. Suppose what happened, extra chromosome is either added to the genome or it is deleted from the, removed from the genome. If it is added, then it is known as hyperploidy, okay, and if it is deleted, then it is known as hypoploidy. Now hyperploidy is again of two types, trisomy and tetrasomy, that is twice n plus 1 is trisomy, okay. It has one chromosome in triplicate. Suppose we are getting a condition twice n plus 1 plus 1, that means two chromosomes are added twice, that means it is a double trisomic condition. Got it? It is not tetrasomy. Remember, here two chromosomes, extra chromosomes are added of two pairs, that's why this is a double trisomic condition. Now this is observed in case of Down syndrome, 21 trisomy, in Patau syndrome, 13 trisomy, Klinefelter syndrome where one extra chromosome is present, okay. So this type of organisms actually they will have 47 number of chromosomes. In case of human being, if we see, we know that the normal chromosome number is 46, isn't it? If the organism is having trisomy, that means it will have 47 number of chromosomes. Got it? Then tetrasomy means twice n plus 2. That means here one chromosome is replicated 4 times. Got it? That means it is represented 4 times. So here, so the chromosome number will be what? 48. Got it? Then if the chromosomes are deleted, then we call it as hypoploidy. It is again of two types, monosomy twice n minus 1 and nullisomy twice n minus 2. If one chromosome is being removed or a homolog consists of only one set, then it is known as monosomic condition. For example, Turner syndrome, which has one less sex chromosome, okay, that means here 45 chromosomes will be present. Nullisomy means here 2 chromosomes are missing, that means the conditions will be 44, the chromosome number will be 44, okay. Next we come to the change in structure of chromosomes. So this is called chromosomal aberrations. Now this aberration may be either intrachromosomal or interchromosomal. Intrachromosomal, that means within the same chromosome. It is divided into 3 types: deficiency, deletion and inversion. If a terminal segment of a chromosome is lost with the help of a single break, then we call it as deficiency, okay. Now here you see, if this is the chromosome 1 2 3 4 5 6, okay, here a single break is occurring for which the chromosome will be 1 2 3 4, but remember this is occurring only one chromosome, okay, of the homologous pair. Again, if a intercalary portion, that means a middle portion is broken out by 2 breaks, then it is known as deletion. If terminal portion, deficiency. If middle portion is lost, then it is deletion. Here you see in the chromosome A, B, C, D, E, F, G, here D and E, 2 breaks are taking place and the resultant chromosome is A, B, C, F, G. Got it? That means D and E has been deleted from here. Inversion means what happened here, the chromosome segment will get inverted by 180 degree. That means this is the chromosomal segment A, B, C, D, E, F, G, okay. It will take the shape of a small e, that means it has been inverted or turned by 180 degree. And so when it is changing, that means the genes present in the chromosome will also alter, isn't it? So for that reason, when it will again break here and then rejoin, it will form a straight chromosome, then the sequence will be changed, that is B, A, B, E, D, C, F, G. Got it? So here actually the whole segment will get changed, it will get inverted by 180 degree. Then sometimes what happened is, now you know what is centromere, isn't it? The point at which the chromatids remain attached. So if the inversion involves the centromere, that means see in this small e, here the centromere is being present, isn't it? So this type of inversion is known as pericentric inversion. But if the centromere is outside the e, that means outside the inverted portion, then it is known as paracentric. Okay. Next is interchromosomal aberrations. Interchromosomal aberrations, that means within two different chromosomes it will occur. For example, duplication. That means here extra chromosomal segment is attached to the homologous pair. Got it? Now see, A, B, C, D, E, F, G. This is the normal segment. Here A, B, C, D, C, D, E, F, G. That means a portion is added in the middle portion. So this is called duplication. Deletion deficiency means loss. Duplication means addition of an extra segment. Next, we come to translocation. Translocation means exchange of chromosomal segment between non-homologous chromosomes. Okay. Now see, if you have remembered, we have studied in case of crossing over that chromosomal segment exchange take place between homologous chromosome. Okay. But here chromosomal exchange is taking place in between non-homologous chromosome. Now see here, 1, 2, 3, 4, 5, 6, 7. This is a normal homologous chromosome pair. A, B, C, D, E, F, G. This is another chromosome pair. But these two chromosomes are actually non-homologous, isn't it? So here what happened? Chromosomal exchange is taking place. Chromosomal segment is exchanged. See here, F, G, and here 6, 7. That means a portion from this is been broken and added to this one, and here also a portion is broken and added to this one. This chromosomal exchange is taking place between the non-homologous chromosome, and this phenomenon is known as translocation. Got it? If homologous chromosome is there, then it will be crossing over. If non-homologous, then translocation. Okay, so this is all about the chromosomal mutation. Now we come to gene mutation. That means the change that will occur in a particular type of gene. It is thus accordingly two type. Either it may be point mutation if the change is occurring strictly in one base pair, and it may be gross mutation if it is occurring in more than one base pair. Now actually gene mutation occurs by inversion. That means the base sequence of DNA segment will be reversed. Now for example, you see here GCC, okay, TAT, TTG. Now these are actually codons, okay, genetic codes which will code for amino acids. Now each of these triplet codons represent one amino acid. Suppose due to inversion these two are altered. Now see, it is TTG, here it is GTT, okay. Again here it is GCC, here it is CCG. That means the whole sequence has been inverted. Okay, and as a result what happened? The amino acid they will code will also alter. Okay, it will be changed. Now next is substitution. That means here one base pair is replaced by another. If a purine is replaced by another purine or pyrimidine by another pyrimidine, then it is known as transition. Now here you see G and A. Now you know what is purine and pyrimidine, isn't it? These are nitrogenous bases. Purine is of two types, adenine and guanine. Pyrimidine is of two types, cytosine and thymine. Now when guanine is replaced by adenine, that means a purine is being replaced by purine, then it is known as transition. Or sometimes what happens? C may be replaced by T. That means pyrimidine replaced by another pyrimidine, then that particular phenomenon is known as transition. But if sometimes what happen, a purine is replaced by another pyrimidine, okay, not by a purine, then it is known as transversion. Suppose adenine, which is a purine, is replaced by cytosine, which is a pyrimidine. So here this particular change is known as transversion. Okay. Then next is tautomerization. Tautomerization means what happened? It is reversible change in the location of a hydrogen atom or molecule, and thus the whole structure will be changed. Now see here, this is a cytosine. Okay, in cytosine, this is the structure of chemical structure of cytosine. Here the amino group is being replaced. Okay, the hydrogen is being shared and the position is altered. Here this hydrogen goes and come attached to the nitrogen, so the whole structure change. Now this is a normal cytosine and this is a tautomeric cytosine. Now due to tautomerization, actually what happened, forbidden base pairs occur. That means if cytosine has to pair with thiamine, that instead of thiamine it will pair with either adenine or guanine. Got it? That means it will change the structure. It will completely, the base pairing will be changed. This so and thus form the forbidden base pair. That is due to tautomerization. Okay. Next we come to frame shift mutation. Frame shift mutation, it involves the insertion and deletion of base pairs. That means what happened here, either a base pair is added or it may be deleted. Now for example, you see here ATT CGC TGA, this is a sequence. Okay, now this is a codon, this is a codon, this is a codon, all are representing amino acid. Suppose we are adding a base G, guanine, to this particular sequence in this position 2. So AGT, now we know that codons always it has to be in a triplet form, isn't it? So what happened, G will enter in next to A, so AGT, then the other T, this T will shift to this side because only triplets are to be formed, so AGT TCG, okay, CGT, so the position will change. That means the frame is being shifted. Got it? So simultaneously what happened, the amino acid will also change. Now then deletion, in deletion also, suppose we are deleting this T, that means from this position ATC, so here from the next base actually it will bring and make the codon triplet form. So this is known as frame shift mutation. That means they are changing their positions and they are simultaneously trying to create a new type of codon, and so mutation is taking place. Got it? So these are all the important types of mutations that are occurring in organisms. And sometimes what happen, actually next we will study hopefully in our next classes about the when we will study about codons, we will get that there are basically actually 3 types of nonsense codons, and these actually nonsense codons, they do not represent any of the amino acids. So when they are present, change is occurring. That means the mutation that is occurring due to these nonsense codon, that is known as the nonsense mutation. Okay, so these are the basic important things regarding mutation. Go through them properly, go through the books, go through the notes, videos. If you have any problem, tell me, because these are actually a bit confusing part also, easy also, not so hard, but still you have to study them properly. Got it? So study them, and if you have any problem, then please let me know.